All right, this is AP Lab 6. This is really two labs we did. We did the PGLOW lab with the transformation of bacteria, and we also did the gel electrophoresis lab where we analyzed DNA, theoretically from a crime scene. Okay? We'll first remind you about the PGLOW and then about the electrophoresis. Okay, the PGLOW lab um, essentially dealt with preparing agar plates. Agar is a gel that bacteria can live on, and some plates had ampicillin in them and some did not. Ampicillin, if you will recall, let me get my pen operational. Uh, Ampicillin, if you'll recall, is, as we can see right there on that plate, is an antibiotic that would kill bacteria unless the bacteria are resistant. So that's kind of an important thing to remember. The way we altered the bacteria. We were given normal bacteria and we wanted to alter them. We wanted to make the bacteria, we wanted to make them antibiotic resistant so they could survive on the plate of agar that had ampicillin on it. So we wanted to make them resistant. And for fun, we also wanted to make them glow in the dark. The way we did this was we gave them we exposed them to a plasmid, which is, if you remember, a small ring of DNA that has kind of accessory genes on it for a bacterium. Bacteria can pick these up from their environment. When they do, that is called transformation because the bacteria may be transformed and have new traits that it did not already possess. So we had a plasmid, a ring of DNA, that had been genetically altered. It had been altered to contain a gene from a jellyfish. Okay? And that gene was the PGLOW gene, the gene that codes for green fluorescent protein. It also had a gene in it in a different location for antibiotic resistance. So if bacteria picked up this plasmid, they theoretically should be resistant to antibiotics, and they also should be able to glow in the dark. To transform these bacteria, if you'll recall, we had to heat shock them, which meant we heated them up and then immediately chilled them down, which makes them more readily accept the plasmid that we put into their little test tube environment. And then we incubated them. So this is showing the transformation taking effect, the heat shock. I forgot to mention we also had to put calcium chloride in. That, again, along with the heat shock, encourages their little pores to open, if you will, to accept the plasmid. Then, once they were heat shocked, we spread them onto the different plates. We had regular LB or Luria broth nutrient agar plates that were plain. We had some that had the ampicillin added to them, and so on. And so we put them all over all the plates, and then we looked to see where we got growth and where we didn't. So you should know what this idea of transformation is when referring to bacteria. You get a foreign gene in the bacteria, and this is the gene we inserted right here. Here's the plasmid, rather. Here's the PGLOW plasmid with its ampicillin resistance gene, the PGLOW green fluorescent protein gene, and we also had something else, an arabinose operon. This is reminding you about that whole concept of the operons, an on switch that will turn a gene on or off. We kind of dealt with that concept as well. The other lab, well, let me go ahead and finish. We'll kind of. Okay. conclusions about that bacteria lab. Now this illustration does not deal with the pGlow, but that's fine. That we should draw is that you can insert foreign DNA into a bacterium by using a vector, a plasmid, and in this case, essentially the ampicillin was a selecting agent. If the bacteria got the engineered plasmid, it would grow on the ampicillin medium, as we can see over here. We had ampicillin and we got growth, but if it did not get the ampicillin gene, resistance gene, it would not grow, okay? So in both of these plates we got growth because there was no ampicillin on the plate. So at any rate, that's kind of a memory of what we did. Now, the electrophoresis, we also want to talk about that. Let me bop you back here a minute. Remember with the electrophoresis, first of all, we cut DNA into fragments with enzymes called restriction enzymes or restriction endonucleases. The enzymes recognize certain palindromic sequences and they will cut the DNA at those specific sites. And then your job, once you have cut the DNA, is to place these DNA samples that have been chopped up into a gel and to allow those fragments to be separated, and we should see different banding patterns because no two people's DNA is exactly the same, and so these restriction enzymes will cut and find those palindromes in different places on people's DNA, and we'll end up with different fragments. And so this is talking about that right here. Remember how gel electrophoresis works. Because DNA is negatively charged, when you turn on the electricity in the gel electrophoresis chamber, here's our chamber. The DNA, if we put it in the gel at this end, is going to be pulled that way towards a positive electrode. We have the negative electrode here. Because DNA is negative, it'll be drawn toward the positive electrode. And of course, the tinier the pieces of DNA, the easier it is to move them through the gel, so they will move faster, and that's how the bands get separated. Yeah, this is pretty much just going over that. Remember, we made the gel using the little comb and whatnot. We have the DNA samples that we cut with different restriction, with the samples, I believe in our case, were from different suspects, and we cut them each with the same enzymes, but we ended up with different banding patterns because they were all different suspects. Only one matched the crime scene evidence. Here you can see a gel. This band must have been the smallest because it traveled the furthest in the given time. This band must have been the largest because it didn't go as far as any of the other ones did, and so on. I am fairly certain that you all did this essay right here, or at least some of you did. This is showing you a band of DNA. And showing you where some different restriction enzymes cut it. Restriction enzyme X cuts in these 3 locations. Y only has 1 restriction site it recognizes, and it's giving you how long the fragments are in terms of base pairs, in terms of how many A to T, C to G pairings we have in each fragment. And this asked you to explain how gel electrophoresis works and then to describe what results you would get in each of these cases. And as I remember, most people did okay with that. Okay. restriction fragment length polymorphism. We've mentioned it. It is essentially what we did. And if you've got questions about that, we can go back during review and catch those. PCR, we never did talk about this, but PCR is simply a way to copy lots of DNA fast and accurately. It's really made DNA science more doable because we can copy DNA and get large samples that we can use to do experiments with. And we will not talk about that. So, at any rate, we can maybe look at some of this essay. Some of this essay I don't think I would really worry about. All right, at this point in the game, at any rate. I do also want to remind you of one thing. Restriction enzymes, I only mentioned those in context of gel electrophoresis, but we also someone, not us in particular, but the company we bought the stuff for, had to use restriction enzymes to engineer our plasmid as well, because remember, the plasmid had to be cut to insert this jellyfish gene into it. So we use restriction enzymes all over the place when we are engineering DNA or analyzing DNA. Okay, so that's a little helter-skelter. I apologize. It's kind of on the fly for me, but hopefully that will jog your memory about the things you need to think about for those biotech labs. All right, that'll do it. And I will catch you at the next lab podcast.